Hey everybody, today we're going to talk about one of the processes with DNA, and that is how we go from DNA to actually making up proteins. We've talked about how our bodies, and really all biology, is made out of two really main things. Water, so you've heard of that, maybe that life's made out of 70 to 80% water, and then a lot of the other parts of it are made out of protein, among other things, but proteins. And proteins have a very specific shape, size, job that they serve. And all of that is controlled thanks to our DNA. It's the set of instructions that make up those proteins. So there's two steps when we go from DNA to actually making the proteins. The first step is what we call transcription. So DNA has all of the instructions to make every single protein in our body. Transcription is a process where we go from DNA into another molecule called messenger RNA. And that's a process that happens inside of the cell's nucleus. Our cell's DNA never leaves the nucleus. That would be like our brain leaving our cranium and saying, hello, stomach, or hello, intestines. No, we want to keep our brain where it is. We want to keep our nucleus and our DNA where it is. So this messenger RNA is kind of the in-betweener. After the messenger RNA strand is transcribed, it then goes to ribosomes. Now, if we remember, ribosomes are the protein-making factories of the cells. But how do proteins know to be made? Where do they go? Well, we just said that they go to the ribosomes, but how does a ribosome know what instructions to follow? Well, that's carried in that messenger RNA strand, and then eventually we get a protein, which is made out of little circles here that we call amino acids. So let's go into a little bit more depth. So DNA is really how we create the proteins that are essential for our survival. And besides water, proteins are arguably the most important with structure, with those enzymes, with reaction centers, and really provide a lot of purposes to the human body and to all biology. So first of all, that DNA molecule we said is a double helix. It's made out of a sugar base phosphate group. Same thing with RNA, but RNA is what we call single-stranded. It has a different sugar called ribose. So RNA is ribonucleic acid, where DNA is deoxyribonucleic acid. So basically it has one less oxygen. It also has a different base called uracil instead of T. So DNA was ATGC. RNA is AUGC. And then RNA is a single-stranded molecule where DNA is that double helix. So this is it in pictures. So here we see that double helix, that twisted ladder, where RNA is just a single strand, different bases, C, G, A, and U instead of the T over here, and then also the different type of sugar, and that's why it's called RNA instead of DNA. Okay, so transcription happens in the nucleus. It is how we get a message from DNA. Remember, the DNA does not leave the nucleus. It's got to have some way of sending a message to the ribosome, and that's where mRNA comes into play. Translation is then taking that mRNA and then making the proteins. So the way that we kind of describe this is that the transcription is like the recipe to the cook, and then translation is the cook making the cake or making the bread. Another analogy we could say, this is the blueprint for the house going to the contractor, and then the contractor actually building the house. So if you like those analogies, certainly use them. Okay, so with transcription, all we're doing is we're going from DNA to messenger RNA. So what needs to happen is there needs to be an enzyme involved that's called RNA polymerase. And what it does is it basically unzips and then figures out what the opposite of the DNA is. Okay, so it's basically what we call the complement, and then also instead of the letter T, remember we're using the letter U. So this molecule, RNA polymerase, does the job that the three enzymes in DNA replication does. It unzips, it attaches bases, and then it zips it back up when it's done. The reason why this can happen is when we go through transcription, we don't copy all of the DNA. We're talking about just a small segment of the total of DNA. So that's why it doesn't necessarily need all three enzymes to make that happen. So once that messenger RNA strand is created, then we go to the next step, which is called translation. So here, we're actually taking that messenger RNA it's going to leave little holes in the nucleus called nuclear pores, and then it's going to dock with a ribosome. And remember, ribosomes are what make proteins. The ribosomes then read or translate that sequence of letters in groups of 3 letters. They're called codons. Each codon corresponds with a certain amino acid. And then there's another molecule called transfer RNA or tRNA that will actually go and get the desired amino acid that's needed. So if we can imagine, this is the mRNA strand, and if we look at our first 3 letters, G, C, U. So what transfer RNA will do is it will say, okay, we got to go get G, C, U. And then we use this chart, can also be a circle chart, but the square one's a little bit better to look at. So we look at it and we see first base G, second base C, and then U. So what that codes for every 3 letters is what's called an amino acid. So GCU codes for the amino acid called alanine. The next one, ACG, would code for threonine. The next one, GAG, would code for glutamic acid. So every 3 groups of letters that you see is going to bring in amino acid. So that's the recipe that's being followed. So we could say, oh, this is for flour, this is for sugar, this is for baking soda, this is for baking powder. And eventually we get that recipe of whatever's needed. So this molecule, messenger RNA, ribosomes is where it happens. And then here's that transfer RNA that I was talking about. So what it'll do is it will read the 3 letters on messenger RNA. It will go and get whatever amino acids needed, and then it brings it back to the ribosome. And here's kind of another picture showing what that might look like. So again, every 3 letters, here's transfer RNA. It goes and gets whatever amino acids needed, brings it to the ribosome. And so what ends up happening is we start to get kind of these, like cards on a train, or we see like, beads on a pearl necklace, and they start to add up. Well, eventually, these will start to bend and kink and twist because of the chemistry that they have. And then we get a full protein, which remember we draw those kind of like globs. And so the making of messenger RNA from DNA, remember we called that transcription. That's just one pretty simple step. And then translation is a little bit more involved where we actually have to go and get them. Now, sometimes we make mistakes. Our cells make mistakes. And remember, don't think of all mistakes or mutations as bad. This is a dramatic one where we're basically taking a duck and we put a wolf on it. That's sarcastic here. Some of these mutations can actually be beneficial, and that's how adaptations, evolution, natural selection can happen. So we kind of break them down based on what happened. So different types of mutations that we can have. A point mutation is where there's just like one spot on the chromosome where a mistake happened. So if there is, let's say, a group of letters where something was missing, we call that a deletion. So if we say that this is our original, and then all of a sudden we're going over here, we see that C and D were lost. Translocation is where they have like switched where they're found. So instead of it maybe being ABC, it becomes AB, X, Y, Z. An inversion is where there's a flip. So we see that, these genes might be present, they just might not be present where they're supposed to be. And in some instances, that's why it doesn't matter. As long as the genes are there, you're okay. But if we have, for example, an inversion and there's maybe like a timing mechanism that's involved, then maybe we have a mutation that could be lethal, or it could be a negative mutation. But again, we don't always think of mutations as negative. So point mutations happen in one particular spot. There's also a group called, and these are oftentimes the ones that are a little bit more severe, but we also have what we call frame shift mutations, where a group of 3 letters might be completely off. So let's take a look at some examples of the frame shift versus the point shift. So let's say that this is our original sequence. Okay, we're going to call this our base sequence. And what we're going to say is that in this first example, we're going to say that this C and G flip, okay, also known as an inversion. So first of all, we want to figure out, okay, what is the original sequence? And this is where we'd use that amino acid chart. So AUG is a very famous one. This is our start codon. GGG stands for the amino acid glycine. CCC stands for the amino acid called proline, and this is our stop codon. All right, so we have a start, a stop, and then two in between. Okay, so what if C and G flipped? Well, AUG is unaffected. The GGG up here now becomes GGC. Well, what's nice is that, okay, that still actually codes for glycine. So we're still good there. But now instead of CCC like we have up here, now we're GCC. This is a different amino acid called alanine, okay? And then here we'd be okay because of stop. So when we just have one thing that's affected, we call that a point mutation. We can literally point at it on the mRNA and say, okay, this is what happened. That's where the mistake is. This other one is a little bit more involved. So let's say, for example, that this C was erased. It's just gone. Okay, there's a deletion. We'd still be okay here. This would still be our start. These three, still okay. That would be our glycine. But now instead of CCC, we have to shift over and this becomes CCU. So this is now a different amino acid called proline. And now we also end up with what we call an incomplete protein. And so we see that originally up here, it was supposed to stop. It was supposed to stop. Well, if we don't run into a stop codon, this thing could go and go and go and go and go on and on and on. So sometimes this is called a run-on protein where the stop is disregarded and just keeps going. But regardless, we collectively call these, whether they stop short or they keep going, we call that a frame shift mutation. So remember, these are supposed to be read in groups of 3 letters. And when that sequence gets messed up, now we might have a whole bunch of either negative or, again, sometimes mutations are positive things that can happen. So point shift, one spot. Frame shift, it might change for a really long time. Those are types of DNA mutations.